Hi, my name is Geoff Oxnard. I am a thoracic oncologist at Boston University and Boston Medical Center, and I am the head of clinical development here at Foundation Medicine. It's my pleasure to speak to you about liquid biopsy, present and future. And here are my disclosures. I am an employee at Foundation Medicine, where we work to develop technologies like liquid biopsy to improve cancer care. And I'm excited to speak to you on some perspectives on where I see this field going and where it could be going next. I'm going to talk about some liquid biopsy basics. I'm going to talk about liquid biopsy for complex variants and liquid biopsy extending past lung cancer. And finally, I'm going to speak to liquid biopsy for ctDNA monitoring, an exciting new future we can't wait to take advantage of in the near term. And so let's get down to the basis of liquid biopsy. What are we talking about here? We're talking about a challenge in oncology where there is a historic precedent of empiric cancer care, where patients get the best possible treatment you could think of. Maybe it's chemotherapy, maybe it's a guess at chemoimmunotherapy, and a future of precision cancer care that is rapidly happening ahead of us, where patients, through finding the right pill, can get the right mutation, can get to a pill, or through finding the right biomarker, can get to an immune therapy, or a PARP inhibitor, and a growing array of precision therapies. And me over my 10-year career, and many of us in the audience, see testing as the catalyst that helps us move patients from this historical precedent of treating based on your best guess to treating based upon a precision diagnostic that steers you in the right direction, hopefully getting patients better outcomes with less toxicity, in a more patient-centered and honestly in a physician-centered way. But doing that can be challenging. And so enter liquid biopsy as one of the ways of trying to do that with fewer barriers, fewer logistical hurdles. And this relies upon the fact that tumor tissue can shed DNA into the blood through apoptosis. Now, of course, healthy cells also shed DNA into the blood. But if you can sift through all of that cell-free DNA, down to 1% or less is tumor DNA. And if you can find that tumor DNA, you can characterize the underlying cancer in that patient and figure out its biology and figure out learnings about how you might be able to treat that patient. Shed is variable and can be impacted by all sorts of things. We have learned that shed varies across cancer types. And on one side, you have glioma here. Brain tumors are low shed cancers. Brain metastases are a low shed phenomenon. You have other cancers like sarcomas with more modest shed. Pancreatic cancer, actually, surprisingly lower shed overall, given the aggressiveness of the cancer. And on the other side of it, small cell lung cancer, where there's exploding with DNA. And lung cancer is on that, non-small cell lung cancer is on that side of the spectrum. It's a relatively higher shedding cancer, though it is variable. This is all shed in advanced cancers. And yet we know the more aggressive a cancer is, with bone mets, liver mets, you can have more ctDNA shed. And if the cancer is more limited in its spread, lung only, lung and brain, you might have less ctDNA shed. But you can't know specifically for a patient until you look. Actually, what's neat about the tools we've been developing, this is Foundation One Liquid CDX, is that it's designed to consider the fact that sometimes you have more shed and sometimes you have less shed. And so when you have more shed, you actually get more coverage the way this assay is designed. It's so creative, right? You get 324 genes when there's enough ctDNA content above 0.5 or 1%. And so the better, richer the DNA in the specimen, the richer signal you get back from the sequencing. But when you are really scraping the barrel to get low-level DNA coverage, low-level DNA levels, you need deeper coverage to get down to that signal. And so for those key 75 cancer genes, when you're down below 0.5%, 0.1%, that's where you need that extra deep coverage to find those key drivers and resistance mutations. And then, of course, when there is no tumor DNA, we know you have to reflex to tumor testing, right? And so a negative liquid biopsy needs to be followed by confirmation with tissue testing, because we know shed can be variable and you don't want to be caught with a false negative result. One way we've looked at shed is to understand how it impacts assay sensitivity. So this is data from AACR. On the left, you see an algorithm that looks at the aneuploidy of germline SNPs. The more aneuploidy, the more shed, the more bouncy the genome is. And you can quantify that. We call that tumor fraction. And when you see elevated tumor fraction, which we define as around 10% or so, that liquid biopsy is so rich with ctDNA, it is practically a tumor biopsy, right? And that liquid biopsy you see on the right, these green bars, has 90% or greater sensitivity for all of these key driver mutations across many different diseases and cohorts. There's MET right there, 100% sensitivity for MET mutations when there is high tumor content. The blue line is the inverse, that when tumor content is not as elevated, you have a risk of false negatives. Your PPA can be impaired. You can still find stuff down at 0.1%, but because PPA can be impaired, the absence of high tumor signal it really reminds us as clinicians that negative can't be fully trusted, and therefore follow that up with tumor testing to confirm that we haven't missed a key driver that might be impactful in this patient. Importantly, shed is also prognostic. High shed cancers, you have to act quickly. You can act on that liquid biopsy. Lower shed cancers can be better prognosis, and it might actually mean you have more time to follow through and get that tissue testing to solve their cancer before starting on therapy. And so blood is compelling, less invasive, rapid. Tissue is the reference truth. You can have confident positives and confident negatives. These are two flavors of how we can get answers for our patients. And so it's important that we, understanding their limitations, we consider using these for the right patient. In that patient where I have a great specimen available, I am going to order tissue NGS. But in that patient where tissue is not available, I'm trying to find it, it is in another lab, it is of borderline quality, I might start with that blood specimen to get that first look at what's going on, understanding that I will follow that negative blood with tissue. These are two tools in our toolbox, and for different patients will help us get that patient to results that can impact their treatment. So what kind of results are we looking for here? And I think it's important that when we use liquid biopsy, I want it to perform beyond the world of EGFR T790M, the world where liquid biopsies began, right? Let's make sure our liquid biopsy can capture the whole spectrum of the cancer genome. This is evidence, like many have shown, that the spectrum of variants you see in liquid can be comparable to the spectrum of variants seen in tissue, where you can see point mutations, complex alterations like rearrangements, amplifications even sometimes, okay? And so we do want our liquid to be representative of our tissue genome with that caveat of sometimes low sensitivity. And the other good news is we know that when you treat patients based upon those liquid results, you get the same great outcomes, right? And that targeted therapy based on a liquid result gives you reliable outcomes as compared to targeted therapy on the tissue result. It's so important that these be actionable and we can use them towards patient benefit. But I also want to trust that they can do the difficult things, right? This is recent data that scientists on my team reported looking at MET exon 14 splice site mutations. And what you see here is the distribution across the MET region, intron 13 to intron 14, of the kinds of variants you can find that end up giving you activation. And what you see is two columns here. The left column is tissue, the right column is liquid. And you're seeing that the relative prevalence is comparable across all this. Tissue, liquid, they are both finding us the same diversity and type of activating MET alterations. I want my liquid biopsy to be reliable, just like a tissue biopsy when it finds them. It can't have gaps. One gap actually I noticed is whole exon deletion. That's the small red bar, a very rare variant that we didn't see in liquid in this cohort. I'm not sure it's impossible, but I'll acknowledge that really big deletions can be harder to see in liquid. And yet you see clearly there that there's a diversity of alterations liquid can identify. And that gives me trust that liquid finds these MET alterations, even though some have argued they can be hard to find in DNA, and yet not the case all the time. Similarly, I need to be able to find fusions in ctDNA. And that's actually one of the first things when I got to Foundation, I said, please show me that we can find ALK fusions. Show me that we can find RET fusions, right? That when we see an ALK resistance mutation, we find the driver. We cannot miss that. And that's the case, right? We find really fantastic sensitivity of fusions, ALK, ROS, RET, the whole complex diversity, seeing them across cancer types. And when there is tumor DNA present, we detect the fusion. And so that gives me a lot of confidence that we can meet a very clear need from liquid biopsy, a very clear need from high quality hybrid capture DNA testing, which is to find the easy variants and to find the complex variants to get those patients treated. And yet we're moving now past lung cancer, right? Here's an example in breast cancer. I don't want to just be finding the common PCRable variants that are the CDX for alpelisib in breast cancer, but a whole diversity of PIK3CA mutations that we can detect, each with a biology highly comparable based upon their genomic features, and even outcomes to say they benefit from targeted therapy. So as we think about PIK3CA and how this target is catching on in many cancer types, I think this diversity of biology we see now in breast cancer makes us revisit our thinking in lung cancer. And maybe some of these actually might be targetable, and it deserves investigation given this complex diversity of ways in which PIK3CA can be activated in our cancer patients. Or in prostate cancer, right? I studied T790M, the resistance mutation, for so many years. And now here to be able to, this is my first paper at Foundation Medicine to describe prostate cancer resistance. What an incredible diversity of ways in which AR can be altered in prostate cancer through amplification, very common, you see in the red here, highly detected in liquid biopsy. This is all liquid biopsy results. Point mutations, complex point mutations mixed together, indels, complex rearrangements, all of this detectable in liquid biopsies and quite reflective of our findings in tissue biopsies also. And finally, complex alterations. This is now looking across the genome at mutational burden. And we're now describing our experience doing this with a over 300 gene panel on liquid biopsy. And you can find the mutational burden. The prevalence in liquid is a little bit lower, you see here, than in tissue of elevated TMB. And yet when your TF is high, your elevated TMB is quite comparable or higher even in tissue. Okay, so you can find a real diversity of mutational burden. When TF is elevated, better correlation between tissue and liquid. When TF is low, less correlation. So really when there's tumor there, we think that this blood-based TMB measure is more representative of the tissue-based TMB. It does require shed to see it. And so when you see elevated BTMB, it may be an aggressive finding, but we are hopeful to now build out that utility story that can be then targeted with immunotherapy as a pragmatic alternative to tissue TMB when tissue is not available, which we know is the case for many of our patients. And so a rich complexity of things we can find in liquid biopsy, a growing array of signals that we can get out of the genome. And that leads us to now, what are we going to do next with this tool? And one thing I certainly learned developing liquid biopsies over these years is that docs want to use them creatively. They want to be able to send them for all sorts of reasons, and we want to empower that through building evidence around using in creative ways like repeat testing on therapy. It's one of the exciting and elusive opportunities with this. Here is data that was reported just recently at World Lung. Here, through a collaboration with Roche, we study serial ctDNA levels. This is on chemoimmunotherapy using a research cohort. And what you see here in the green line is that patients having a response have more dramatic decrease as opposed to patients with stable disease or disease progression, right? And we sort of know that, right? That the ctDNA can correlate with imaging, but can it provide more than imaging even? And so here in an analysis, we take patients who objectively at cycle 3 after 2 rounds of chemoimmunotherapy had stable disease. And if you then separate that stable disease group here in the middle by ctDNA, the patients who have radiographic stable disease but have clearance of their ctDNA have more favorable outcomes, like those patients who've had a PR. It is an alternate measure of response, not seen on imaging, but seen in the blood. And those patients who have stable disease but do not have ctDNA clearance, their outcomes are much more like patients with disease progression. I'm not ready to call it necessarily full progression, but certainly this is more ominous. And so the idea that in all those situations where imaging is failing us, and I think you and I know in the clinic, there are plenty of those situations where imaging doesn't feel like it's telling the story. ctDNA has this opportunity to dynamically add to the story and clarify the outcome for a patient. So really exciting. How do we get this kind of tool into the hands of docs so they can use it? Here is another analysis. This analysis uses now Foundation One liquid NGS and measures tumor fraction across the genome. And in measuring tumor fraction across the genome, we can actually see this is now a correlation between increase or decrease in that tumor fraction measurement versus possibility of response on imaging. So clearly a relationship between what we're seeing in the blood and what we're seeing on the imaging, now using a clinical grade liquid biopsy NGS test. And so where do we go next? I want to be able to do this for patients, but as you think about the multiple time points, we need a scalable tool. And that's where I'm more excited about these more nimble, bespoke approaches where you build off a tumor test, you figure out their mutations, and then you test that over time to see, is it going down, is it going up? You test for a personalized set of mutations. You can test that over time. And so we're excited to build into this world with a new technology called F1 Tracker. It's a collaboration with Natera, where you take tissue NGS, F1 CDX at baseline, regulatory grade, profiling, and you then can build off of that a tracking tool, which in advanced cancer can tell you if that patient with immunotherapy is benefiting or not, or even in early disease can tell you if that patient has residual tumor above or below that MRD time point. So really exciting that this is where the future of liquid biopsy is going, and excited to make this a reality for patients. Liquid biopsy, who are those patients we think that can help? In a patient where traditional testing is impractical, infeasible, that's a great patient for a liquid biopsy where that tissue is not available. In a patient with progression or resistance, certainly an obvious place now in lung, breast, prostate, where we can figure out how the genome has evolved. And in those patients where we have uncertainty about what their imaging is looking like, can monitoring add to the story? Really excited to build into this world ahead of us. We just need the tools to do it, and looking forward to partner towards making those solutions a reality. Thanks for your time, and we'll be taking questions when we meet next. Bye.